بسم الله الرحمن الرحيم. In this lecture we will talk about a new bacterial genera. It is the Mycobacterium. This genus contains numerous species that can cause a wide variety of chronic infections in humans. We have two most important diseases caused by this genus. A, we have the tuberculosis, chronic, in form of chronic lung infection, and the other disease is the leprosy in form of disseminated infection. Bacteria in this genus retain dye, carboxyl stain, even after we decolorize the slide with a strong decolorizing agent like the acid alcohol. So, bacteria in this genus called acid fast bacilli. The name acid fast bacilli or AFB refer to members of this genus because bacteria in this genus retain stain, carboxyl stain, even after decolorization with a strong decolorizer agent, the acid alcohol. Mycobacteria not stained with the gram stain. Mycobacteria do not stain with the gram stain, but need special staining method. The staining method used to stain this bacteria is called acid fast stain. So in this slide we must know two things. Bacteria in this genus is called AFB, and the stain used for the staining of this bacteria is called acid fast stain. Classification of mycobacteria. How we can classify mycobacteria. Mycobacteria can be classified as following. 1. Mycobacterium tuberculosis complex. In this group we have 3 species under Mycobacterium tuberculosis complex, 3 genus: Mycobacterium tuberculosis, Mycobacterium bovis, Mycobacterium africanum. Tuberculosis, the chronic lung infection in a human, mainly caused by Mycobacterium tuberculosis. A human infection can also caused by Mycobacterium bovis from the cattle, whereas Mycobacterium africanum coming from monkey. The second group of classification of mycobacteria, we have the Mycobacterium other than tuberculosis, MOT, اختصارا abbreviated as MOT, Mycobacterium M, other than O, anti-tuberculosis. Or this group is called tuberculoid bacilli. So in this group, MOT or tuberculoid bacilli or the atypical mycobacteria. These three names, MOT, atypical mycobacteria, tuberculoid bacilli, refer to 4 species. Mycobacterium avium intracellulare complex, Mycobacterium kansasi, Mycobacterium sacrofolacum, and in the 4th species we have Mycobacterium marinium ulcerans. Marinium ulcerans. Mycobacterium marinium and the Mycobacterium ulcerans. In Mycobacterium avium intracellulare complex, this type of mycobacteria, widely present in the environment and animal, namely the birds. This type of mycobacteria cause opportunistic infection. This type of mycobacteria cause infection, opportunistic infection in immune-compromised patients. The lungs primarily affected by this type of mycobacteria. Then the infection spread to involve other organs of the body, causing disseminated infection like that occur in AIDS. So in AIDS patient, the mycobacteria causing the infection is the Mycobacterium avium intracellulare complex. This type of mycobacteria resistant to the treatment. We have special type of anti-tuberculous drug used to treat infection caused by these bacteria. And in this group, the avium intracellulare complex, resistant to the first group of anti-tuberculous drug. So we use combination of a drug, namely new macrolides and ethambutol, in treated this infection. 2. The second type of mycobacteria included under the MOT, atypical mycobacteria, tuberculoid bacilli, is the Mycobacterium kansasi. Here we have a reservoir in this mycobacteria, either the wild or domestic animals. In Mycobacterium kansasi, we have a reservoir, wild or domestic animals. Communicability in this type of mycobacteria is low or absent. Pulmonary infection or the disseminated infection caused by this mycobacteria is indistinguishable from the tuberculosis. This type of mycobacteria is susceptible to the usual anti-tuberculosis drug. That's to say, it is not resistant to the first-line anti-tuberculosis drug. Third mycobacteria included under MOT is the Mycobacterium sacrofolacum. Mycobacterium sacrofolacum. This type of mycobacteria found in the soil cause chronic cervical lymphadenitis, mainly in children, rarely cause other granulomatous infection. So the special thing about Mycobacterium sacrofolacum is found in the soil, cause chronic lymphadenitis, usually do not cause other granulomatous disease. The treatment of infection caused by Mycobacterium sacrofolacum is by surgical excision. Excise the involved lymph node by the infection with this bacteria. Again, this mycobacteria is resistant to anti-tuberculosis drug. Fourth mycobacterium included under MOT, we have Mycobacterium marinium, Mycobacterium ulcerans, grouped together because they have similar phenotypic characteristic. These Mycobacterium marinium, Mycobacterium ulcerans, found in water, grow in lower temperature, 31 degrees centigrade, causing superficial skin infection. So, swimming pool granuloma or ulcer of the skin caused by infection with Mycobacterium marinium ulcerans. Treatment of this infection caused by this Mycobacterium group is by surgical excision, tetracycline, rifampicin, ethambutol. The fourth group of MOD is the Mycobacterium marinium ulcerans, found in water, grow at lower temperature, cause ulcer, swimming pool granuloma. The treatment by surgical excision. Sometimes we use tetracycline, rifampicin, ethambutol for the treatment. The third group of mycobacteria, we have the rapid growers. Rapid growers microbacteria, we have the Mycobacterium fortuitum chiloni complex. Again, two Mycobacterium fortuitum, Mycobacterium chiloni included under rapid grower in form of Mycobacterium fortuitum chiloni complex. This group, the rapid growers, free living, found in the soil, in water. From the name, they are rapid growing bacteria, need 3 to 6 days to grow, because mycobacteria usually need around 6 weeks to grow. So the group rapid growers mycobacteria is an exceptional for this rule. That's to say, need 3 to 6 days to grow, whereas the other mycobacteria need longer incubation time for growing, around 4 weeks. Rapid growers rarely cause disease in a human. Rapid growers resistant to the usual anti-tuberculosis drug, respond to amikacin, deoxycycline, cefoxitin, erythromycin, and rifampicin. There is resistance to the treatment with anti-tuberculosis drug in this group. This group can be treated with amikacin, doxycycline, cefoxitin, erythromycin, and we can use rifampicin. Fourth group of mycobacteria is the saprophytic mycobacterium. This group of mycobacteria, saprophytic mycobacterium, free living, not associated with a human disease. In this group, saprophytic mycobacteria, if we ask about saprophytic mycobacteria, we have Mycobacterium phylum, Mycobacterium phylli, Mycobacterium cordoni, Mycobacterium smygmatus. 3 species of mycobacterium included under free living saprophytic mycobacterium. Mycobacterium fili, Mycobacterium cordoni, Mycobacterium smygmatus. These three species not associated with a human disease. First two species, Phylum, Fili and the cordoni, present in plants, water, soil. Fili, cordoni, present in plants, soil, water. The third species, smygmatus, present in sebaceous secretion of a human. The fifth group of mycobacteria is the Mycobacterium leprae. Its a strict pathogen cause leprosy. This was about the classification of mycobacteria. The mycobacteria can be classified into Mycobacterium tuberculosis complex group, Mycobacterium other than tuberculosis, the atypical mycobacteria, the moth, and the rapid grower mycobacteria, اللي هي exceptional for the rule of having long incubation time, اللي هي rapid grower. The fourth group هو saprophytic mycobacteria, and in the fifth group we have the Mycobacterium leprae. Now the microscopical appearance. How the mycobacterium appear under the microscope. If we look under the microscope and we take the species Mycobacterium tuberculosis, this bacteria need special stain. As we said in the first slide, the stain used to stain mycobacteria is called the acid-fast stain, and one method of the acid-fast staining is the Ziehl-Neelsen technique. So, the Ziehl-Neelsen stain used for the identification of the acid-fast bacilli. Tubercle bacilli in a slide stained with Ziehl-Neelsen technique appear red, slightly curved, slender rods. So, the AFB, the acid-fast bacilli, the tubercle bacilli, the same thing. If we call AFB, acid-fast bacilli, tubercle bacilli, is the same for the mycobacterium. It appears red, slightly curved, slender rods, dimension, length 2 to 4 micron, width 0.2 to 0.4 micron. The background of the slide stained with the Ziehl-Neelsen staining technique is blue. These rods, the AFB, either appear as discrete rods or aggregating together. The AFB, the acid-fast bacilli, the tubercle bacilli, either appearing discrete, far away from each other, or aggregates coming together. This aggregation form a long arrangement. This long arrangement of the acid bacilli, of the acid-fast bacilli, is called the serpentine cords, in which the acid-fast bacilli aggregate and form their long axes parallel to each other. Only the virulent strain of acid-fast bacilli arrange in form of serpentine cords. So, In this slide, we describe the microscopical appearance of mycobacteria. The mycobacterium, or the acid-fast bacilli, or the tubercle bacilli, appear in Ziehl-Neelsen stain slide red. The AFB is a slender rods, slightly curved, red bacilli over a blue background. These AFB either appear as a discrete متباعدة عن بعض or aggregates in form of, we call it serpentine cords. The serpentine cords only occur in virulent strain of mycobacterium. Okay? Now we come to the cultivation of mycobacteria. Mycobacteria need special media for their growing because this genus is a special genus, so the cultivation need a special media. We can group the media used for cultivation of this bacteria in form of 3 groups. First group we call the agar-based media. First group of media used for cultivation of mycobacteria is called the agar-based media. The example of the agar-based media is the Middlebrook 7H10 and the Middlebrook 7H11. These two are examples of agar-based media used for the cultivation of mycobacteria. Special lab referred as a Middlebrook lab, and the code for this combination of the media used for the cultivation of mycobacteria is 7H, either 10 or 11. In this media, in the agar-based media, we have salt, defined salt, vitamins, other cofactors, oleic acid, albumin, glucose, glycerol, and in case of 7H11 Middlebrook, we have an additional the casein hydrolysate. So in the agar-based media contains salt, defined salt, vitamins, cofactor, oleic acid, albumin, glucose, glycerol. وبحالة ال 7H11 additive we have the casein hydrolysate. The advantage of the agar-based media, the use of Middlebrook 7H10 and 7H11 in the cultivation of mycobacteria for 1. Observation of colonies morphology. If we want to study the morphology of colony of different species of mycobacteria, we use the Middlebrook, the agar-based media. 2. Used to test the susceptibility of the mycobacteria to different anti-tuberculous drug. 3. If we add antibiotics to the agar-based media, this media can be used as a selective media for the cultivation of mycobacteria. Disadvantage of the agar-based media, the Middlebrook 7H10 and 7H11, it cannot be used for the primarily isolation of mycobacteria because the agar-based media are less sensitive for the cultivation, for the primarily cultivation of mycobacteria. That is to say, Middlebrook media need high inoculum size to be grown as a culture of mycobacteria, so cannot be used as a a primarily isolation culture of mycobacteria. تحتاج إلى كمية كبيرة من الأسيد فاست باسيلاي متوافرة في السبيسيمن in order to appear as a culture, as a colonies on these media. فلذلك ميدلبروك ما تستخدم للعزلة الأولى للمايكوبكتيريا. تستخدم الادفانتج ما اللي ذكرناها بالنقاط الثلاثة الواضحة أمامكم. Second kind of media used for cultivation of mycobacteria is the egg-based media. The egg-based media, the example of egg-based media, we have the Lewiston-Jensen medium, LJ medium. The LJ medium or the Lewiston-Jensen medium is an example of the egg-based media. The egg-based media is a second type of culture media used for cultivation of mycobacteria. Again, in this group of culture media contain defined salt, glycerol, and a complex organic material in form of fresh egg yolk, and the potato flour. إضافة إلى other combination in various concentration. So in the egg-based media we have a complex organic material in form of a fresh egg كاملة أو جزء منها اللي هي الegg yolk مضاف له potato flour إضافة إلى other combination. In this media, in the Lewiston-Jensen media, we have a malachite green. الملكايت green, صبغة الملكايت green included in the LJ medium to inhibit other bacteria, to inhibit the growth of other bacteria present in the sample. So by this, the Lewiston-Jensen medium, the egg-based media, can be used as a media for the primarily isolation of mycobacteria. So the advantage of the Lewiston-Jensen medium, it's used for the primarily isolation of the mycobacteria. Why? That is because this culture medium contain malachite green dye. This dye inhibit the growth of other bacteria present in the sample. The egg-based media again can be used as a selective media by adding other antibiotics in the combination of the media. So the second group of media used for cultivation of mycobacteria is the egg-based media. With example of the egg-based media, we have the LJ medium, the Lewiston-Jensen medium. In this medium, إضافة إلى defined salt والglycerol, we have the complex organic material in form of fresh egg or egg yolk plus flour, potato flour. In this media, we have a malachite green dye. This dye inhibits the growth of other bacteria, so this bacteria used as a media for primarily isolation of mycobacteria. Third group of media used for cultivation of mycobacteria is the broth media, the Middlebrook 7H9 and the Middlebrook 7H12. This media, the broth media, used for biochemical testing of mycobacteria, to test the biochemical test on mycobacteria, the presence of special enzymes or resistance to certain chemicals. In Middlebrook 7H12, the growth of mycobacteria is detected automatically by the presence of 14 radio-labeled radio-labeled palmitic acid. So the advantage of Middlebrook 7H12, this media, the growth of mycobacterium is detected automatically by the machine. This is because the carbon source is a radio-labeled, radioactive carbon 14 palmitic acid included in this media. So once the mycobacteria grow in the Middlebrook 7H12, utilize the radio-labeled carbon present in the palmitic acid, releasing the radio-labeled CO2, and this radio-labeled CO2 is detected by the machine. By this, positive culture can be detected in 2 weeks. So in this group, the broth media, we have two examples, Middlebrook 7H9 and the Middlebrook 7H12. These media used to study the biochemical testing of the mycobacteria. In case of Middlebrook 7H12, the advantage in this media, in this special medium, the Middlebrook 7H12, the carbon source is a radio-labeled carbon, leading to the growth of mycobacteria to be detected by the machine, and the incubation time, it will be lower to 2 weeks in order to detect the growth of mycobacteria. Now we come to the growth characteristic of mycobacteria. In the growth characteristic, we will talk about the growth rate and colony morphology. The growth characteristic of mycobacteria in general, it's an obligate aerobes, strict aerobes, need an increase in the CO2 tension to enhance their growth. A growth rate of mycobacteria much slower than most bacteria. The growth rate أبطأ من بقية البكتيريا. And they need a prolonged incubation at 37 degrees centigrade up to 8 weeks. Due to the slow growth rate of mycobacteria, mycobacteria in general need a prolonged incubation at 37 degrees centigrade up to 8 weeks. That is to say, we cannot discard ما نخرج نتيجة أنه negative for mycobacteria إلا بعد ما نبقي العينة مزروعة على وسط الزرع المخصص للمايكوبكتيريا لمدة 8 weeks. The saprophytic mycobacteria is an exceptional. It needs to grow more rapidly and in less than 1 week. Now, according to the growth rate, if we use a growth rate as a point of dividing the mycobacteria, we have the rapid growers mycobacteria and the slow growers. Rapid growers mycobacteria that need less than 7 days to grow, whereas the slow growers mycobacteria need more than 7 days to grow. Under the rapid growers, we have the free-living saprophytic mycobacteria اللي هي exceptional for the rule, and the mycobacterium fortutum chiloni اللي كانت included and the rapid growers, a group of classification of mycobacteria. So, In the rapid growers, the mycobacterium that need less than 7 days to grow, we have the saprophytic mycobacteria, the free-living mycobacteria, and the rapid growers group اللي هي mycobacterium fortutum chiloni. The slow growers, the mycobacterium that need more than 7 days to grow, it's the usual mycobacteria اللي هي ذكرنا إنه all the mycobacteria share the characteristic of being a slow growth rate and need a prolonged incubation. Under the slow growers, we have the Mycobacterium tuberculosis as a prototype. This Mycobacterium tuberculosis need 2 to 3 weeks to grow. The growth characteristic of Mycobacterium tuberculosis, it's need 2 to 3 weeks to grow. The growth of Mycobacterium tuberculosis described as tough, rough, creamy colored, easily detached from the surface of the media, and difficult to be emulsified. This growth characteristic of Mycobacterium tuberculosis, tough, rough, creamy color, easily detached from the surface of the media, difficult to be emulsified, is called the eugenic growth. So the growth characteristic of Mycobacterium tuberculosis in the media is described as eugenic growth. That's to say it's tough, rough surface, creamy color, easily detached from the surface of the media. بسهولة ينرفع من سطح الميديا، بس من تيجي تعمل له emulsification على السلايد it's difficult to be emulsified. Eugenic growth. In case of Mycobacterium bovis, need 5 to 6 weeks to grow. The growth characteristic of Mycobacterium bovis, smooth surface, adherent to the surface of the media, easily emulsified. These three characteristics of the growth of Mycobacterium bovis is called the dysgenic growth. So the characteristic growth of Mycobacterium bovis is dysgenic growth. Smooth surface, adherent to the surface, and easily emulsify. In case of Mycobacterium other than tuberculosis, the multigroup, the atypical Mycobacterium, the tuberculoid bacilli, we can differentiate the growth of this group of Mycobacterium according to the pigment production in relation to the light exposure. If we want to study the growth characteristic of the multigroup of Mycobacterium, the atypical Mycobacteria, the tuberculoid bacilli, this group of Mycobacteria can be differentiated according to the pigment production after a prolonged light exposure. So we have the photochromogenes, scotochromogenes, and non-chromogene. These three groups, the photochromogene, the scotochromogene, and the non-chromogenes, is a group according to the pigment production of Mycobacteria after a prolonged exposure to the light on the media. In case of photochromogene, it gives pigmented growth after a prolonged exposure to the light. This pigment in form of lemon yellow to bright orange. In this group, we have the Mycobacterium kansasi, Mycobacterium marinum, and Mycobacterium cepi. So the Mycobacterium kansasi, Mycobacterium marinum, Mycobacterium cepi is a photochromogenes give lemon yellow to bright orange growth after a prolonged exposure to light on the media. In case of scotochromogenes, these mycobacteria are pigmented in the dark, and the pigment usually deep yellow to orange. Once this growth of the mycobacteria exposed to the light, it becomes darkened to orange or red. And in this group we have the Mycobacterium sacropholacum, Macrobacterium suzelgi, and the Macrobacterium cordoni. The scotochromogenes group of mycobacteria, it gives pigmented growth after incubation in the dark in form of deep yellow to orange growth, and once this growth is exposed to the light, change to deep orange or red color. In this group, the scotochromogenes, we have the Mycobacterium sacropholacum, cordoni, suzelgi. Non-chromogenes, these mycobacteria do not develop pigment on exposure to light. Example of non-chromogene is the Mycobacterium avium intracellular complex and the Mycobacterium ulcerans. In this slide showing the eugenic growth of Mycobacterium tuberculosis on the LJ media with the characteristic rough surface of the growth and the creamy color. In this slide is the table for the classification of non-tuberculous mycobacteria. If we want to classify the non-tuberculous mycobacteria, we have the Ranion group of classification, Ranion group. The first Ranion group of non-tuberculous mycobacteria, we include the Mycobacterium kansasi, Mycobacterium marinium, and Mycobacterium simi. It's a photochromogen. Group 2 Ranion اللي هي المايكوبكتيريوم sacropholacum, suzelgi, cordoni اللي هي scotochromogen. الجروب 3 Ranion اللي هي Mycobacterium avium intracellular ulcerans اللي هو non-chromogen. جروب 4 Ranion اللي هي rapid growers اللي هي Mycobacterium fortutum chiloni. So in this slide is a table of classification of non-tuberculous mycobacteria according to Ranion group. Group 1 Ranion is the photochromogen, Group 2 Ranion is the scotochromogen, Group 3 Ranion اللي هو non-chromogen, and the fourth is the rapid growers اللي هي Mycobacterium fortutum chiloni. So if we ask about Ranion grouping of a classification of non-tuberculous mycobacteria, we have 4 groups. الون هو الفوتوكروموجين، التو اللي هو السكوتوكروموجين، الثيرد هو النونوكروموجين, and the fourth is the rapid grower. In the photochromogenes we have the kansasi marinium ulcerans, simi, the kansasi marinium simi, and in case of the scotochromogen is the sacropholacum suzelgi cordoni, and in the non-chromogen we have the avium intracellular ulcerans, and the fourth group is the rapid grower اللي هي fortutum chiloni. Reaction to physical and chemical agents. Mycobacteria resistant to chemical agents due to the hydrophobic nature of the cell surface and their clumped growth. Mycobacteria تكون resistant للchemicals نتيجة ال hydrophobic surface of the bacteria and the clumped growth. الداي اللي قلنا عليها الملكات green or antibacterial agent البنسلين can be incorporated into media without inhibiting growth of tubercle bacilli. يعني الصبغات الملكات green والأذر antibiotic البنسلين تأثر لي على بقية البكتيريا بس ما تأثر على المايكوبكتيريا. Mycobacteria can survive in acid and alkalis. These can be used to decontaminate the specimen sent to isolate the acid-fast bacilli. And lastly, the tubercle bacilli are resistant to drying and survive for long periods in dried sputum. Now the constituents of a tubercle bacilli. مكونات التوبركل bacilli، مكونات الأسيد فاست bacilli. A. We have the lipids. Cell wall of Mycobacterium, acid fast bacilli rich in lipids in form of A. Mycolic acid, B. Waxes, C. The phosphatides. الليبيدز الموجودة بالسيل وول، السيل وول of Mycobacteria يكون غني بالليبيدز in form of mycolic acid, waxes and the phosphatides. المايكوليك acid هو long chain fatty acids بنوع carbon 78 to 90. This high lipid contents of the cell wall of Mycobacteria is responsible for the acid fastness of this bacteria and inability of this bacteria to be stained with the usual gram stain. One type of mycolic acid present in the cell wall of Mycobacteria is the 3-hallose dimycolate اللي اسم الآخر اللي هو هذا السبيشال kind of mycolic acid موجود بالسيل wall of Mycobacteria. ال 3-hallose dimycolate هو الكورت فاكتور. الكورت فاكتور هو responsible for characteristic growth of virulent strain اللي قلنا علينه السيربنتاين كورتس، وكمان يعمل لي inhibition للmigration of leukocyte and leading to formation of chronic granulomas. So one special kind of mycolic acid اللي هو ال 3-hallose dimycolate اسمه الكورت فاكتور هو اللي يعمل لي growth of the virulent strain of form of serpentine cords. والشغلة الثانية اللي يعمل لي اللي هو يمنع لي migration of leukocyte ويؤدي لي إلى formation of chronic granulomas. The second constituent of a tubercle bacilli هو البروتينز. خلي بالنا إنه each type of Mycobacterium تحويلي على several proteins اللي هي تعمل لي التيوبركلين reaction. The third constituent of a tubercle bacilli اللي هي polysaccharides. Mycobacteria contain a variety of polysaccharides اللي هي تعمل لي induce the immediate type of hypersensitivity reaction. فإذا سألنا على constituent of tubercle bacilli هي الليبيد، البروتين، البوليساكرايد. الليبيد اللي هي ذكرناها أمامكم والأذر البروتينز and the polysaccharides. وأنا أشكركم على حسن الإصغاء والاستماع. تحياتي.